of nanocytometry to cancer research. Thank you, Vadim.
All right. So welcome. What I will, I guess, give is I'll give a quick overview of what Nanocytology Core does, what kind of services it can provide, what kind of resources are available. Let me, as an ansatz, let me just start that when we think about cancer hallmarks, we can think about functional or phenotypical, so to speak, hallmarks, which is phenotypical meaning what cancer cells or cancers actually do. And in both cases, whether we talk about function or phenotypical hallmarks, imaging becomes really paramount. And we can think about this as you have tumor marker environment hallmarks, and you also have the hallmarks relevant to the cells themselves, so subcellular processes, intracellular processes. So the imaging that the Nanocytometry Core provides can really address quite a few of these phenotypes, actually pretty much most of them, with the exception of it can measure gene activity, at least not just yet directly. And let me start with one quick physical fact. Optical microscopy is, I would say, the ideal tool to measure cell or tissue function and structure. The problem is the resolution. The conventional microscopy can't really see structures below what is called the diffraction limit, which is a few hundred nanometers. Just to give you an idea, well, imagine you have the structure of the chromatin which you want to image. That's how it really looks like. With the conventional microscopy, what you would see is something like this. So essentially kind of blurred, myopic image of the structure. Now, recently, and this was actually Nobel Prize in 2014, super resolution microscopy was developed. A number of these techniques are available through a number of facilities here at Northwestern, including the microscopy core. I guess one of the more useful techniques is called STORM, Stochastic Optical Reconstruction Microscopy, which what it does is uses fluorescent labels, special fluorescent labels the structures that you want to image, and then they blink. The fluorescent molecules blink. That's what they do. And at any given time, you have just one fluorescent molecule that is shining brightly, so you can very accurately pinpoint the location of this molecule. And eventually, when lots of them, you can make lots and lots of these images, and eventually can reconstruct the whole thing. In most cases, STORM has resolution down to about 20, 30 nanometers. In exceptional circumstances, it can go down to 10 nanometers, but conventionally it's more like, well, the commercially available instruments go down to about 40 nanometers. More of the advanced instrumentation, some of it has been developed here. It can go down to 20 nanometers. It's essentially you beat the diffraction limit by an order of magnitude or more. But let me just point out one thing, that in many applications, especially when we talk about cancer hallmarks, that's where a label for imaging becomes would be quite useful, and there's a few reasons why. For one thing, the issue with labels is if you work with live cells or live tissues, you can't label everything. In order to have resolution of, let's say, 10 nanometers, you have to label at least, in the worst case scenario, at least every 5 nanometers. If you want to go even further, if you want, let's say, to understand, for example, how chromatin structure looks like, or if you want to image some of the, let's say, protein translation processes, you have to go down to individual nanometers, which means that pretty much you have to put a label in each and every major groove of DNA, and intercalating agents do that. Well, they kill the cells at the same time. So the problem is, especially for very high resolution, you can't with labels do it non-invasively. You're going to perturb the cell function. And the problem with that is if you image that, the question is, which processes are you really imaging? Okay, that's one issue. So the label density. So then you can't use very high label density, and when the label density is not high enough, that's what you're going to get. Imagine you have letter N, you label it, and it may look like you only see the labels, right? So you may see letter H instead. That's just one example. Another thing is that there's always Brownian motion, thermal motion of the labels, which really gives you a little, sometimes gives you a different perspective of what the real structure is. For example, because of this links microtubules in most cases, even though they're, let's say, 20 nanometers, may appear much larger just because of that. You would be incorrectly measuring sizes. Imagine you want to image this, let's say, protein DNA fiber, or understand if it even exists. You're looking for a 20 nanometer fiber. It would be hard to do this using labels because of this. And finally, somehow, PowerPoint is stuck. Okay. 1 second. Okay, we're back. All right, so the approach that Nanocytometry Core took is essentially build a platform of optical technologies with a trick. It combines spatial resolution, which doesn't require labels, with these essentially spectroscopic sensing. So the idea here is that for each resolution voxel, we can acquire some sort of spectroscopic data and measure processes which otherwise would not be visualizable directly. And using this approach, you can go below the diffraction limit. And we leverage a number of technologies developed here at Northwestern in collaboration, as a collaboration among a number of different investigators. And I'm going to give an overview of what kind of technologies are available right now. If you want to look at the most zoomed-in level of the cell, the technology that was recently developed as a collaboration of two labs, Hao Zhang's lab, biomedical engineering lab, and in my lab, is called intrinsic contrast STORM, or IC STORM. So I just told you what STORM does. In this case, in IC STORM, you don't have to use labels. You can achieve 5 to 6 nanometer resolution, and then the sensitivity is further increased about 25 times, essentially going down to individual, potentially monomers through the spectroscopic sensing. At a little higher level, there is a partial wave spectroscopic microscopy, also known as nanocytology, which can image hundreds of cells at the same time. We're talking about much larger, hundreds of microns field of view, with the resolution going down to a few hundred nanometers. It's also a very, very powerful technique to image cell dynamics, essentially intracellular dynamics, with the speed which is measured in, the rate which is measured in millisecond rate, basically. And the sensitivity of this technique is down to 20 nanometers, and in addition to that, it's a very powerful tool to image even very subtle dynamics at the nanoscale. As you know, I will explain later today, even a few nucleosomes move in the cell nucleus, you will be able to sense that, even though the movement itself may be very, very subtle. Let's imagine nucleosome moves plus minus 10 nanometers and doesn't go anywhere else. You can sense this. You can sense this kind of motion. So very powerful as for dynamic measurement, live cell measurement. At a higher level, we're talking about microscopic tissues several microns in size. So we're talking about, if you, for example, there's a need to image a QVEC model, or even a tissue from or a tissue model which is several millimeters across, or matrigel model, or even tissue from an animal. That's where inverse spectroscopic optical coherence tomography, or ISOCT, is available as well. It gives three-dimensional image of a tissue down to a few hundred microns, potentially to a few nanometers, a few microns, with the resolution down to a few microns. Within each resolution voxel, you can sense down to about 40 nanometers. So I'm going to give overview of these three things. Let's start with the ISO-CT. As I mentioned, three-dimensional imaging that gives you generates images like this. This is the tissue. You can go down to, let's say, down to a millimeter or so. In this case, resolution is 10 microns. It can be made better, about a few microns in size. You can do live tissues, and you don't have to use labels. One of the power of the technique is to study extracellular matrix and cancer microenvironment in general. Just give you one example. One of the aspects of tumor microenvironment is the cross-linking of extracellular matrix, such as collagen matrix. And one way to do it, it's if we could, I would probably see where turn on the lights a little. Oh, I don't really see it, but it's okay. So one way existing way how you can image extracellular matrix is to use STORM again. You label collagen fibers. You come up with the generate images like this, which takes several hours to generate actually. And then you try to find features of the cross-linking by calculating mass density correlation function, which informs you about the cross-linking as there's a shift towards larger length scales in this correlation pattern. And you can measure this as through a, let's say, fractal dimension of the structure. The higher fractal dimension indicates cross-linking. That's one way to do it. At the same time, you can do the same thing with ISO-CT within speed that is much faster than a second. And you can also scan millimeter-sized areas to get a very comprehensive picture. So it's a very fast way of measuring ECM cross-linking. In collaboration with the Lonnie Shea, for example, we're able to image changes in cross-linking of matrix cross-linking as cancer cells arrive to the metastatic niche. And for example, as few as 15 cells was enough to have significant changes in the matrix, which could be detectable using this approach. Like I said, you can also use this in live tissues as shown here, or excised ex vivo tissues as shown here. These are images of microscopic biopsies from patients with and without neoplasia. You can also observe changes in the ECM in this kind of tissues. Another powerful aspect of this technique is measurement of microvasculature. And what essentially it does, it measures a number of things. You have sensitivity or images of essentially individual capillaries, all the way down to individual capillaries. This ISO-CT technique can measure oxygen saturation, can measure blood flow through these capillaries or larger blood vessels, or the metabolic rate around each and every capillary. This is actually this technique is available now, but it was originally developed again through a collaboration, three-way collaboration. Professor G.E. at Boston University housing here at Northwestern in my lab. So as you can see here, you can see individual blood vessels, and you can essentially color out these blood vessels. In this case, is the image from the retina. This is the mouse ear. You can essentially color them out based on, let's say, how fast blood flows, or what the metabolic rate is, or the oxygenation of the blood. A few more images, you see this beautiful pattern of vasculature. As it currently stands, ISO-CT is the world's most advanced microangiography technique that exists today, like I said. Very, very sensitive. So now let me switch gears a little bit and show this figure. This actually I borrowed from these are two histology slides. These are normal bronchial cells. These are cells of lung carcinoma. What I wanted to point out is this structure of the chromatin. Smooth appearance here, highly heterogeneous clumped appearance of chromatin in cancer cells. This is something that pathologists use every day, and it's a feature of essentially each and every type of cancer with a very, very few exceptions, essentially no exceptions really. Still remains the most significant, most diagnostic marker of malignancy, and has been for probably 100 years now. So this is just to illustrate how significant the chromatin structure or epigenetic processes in general, how significant they are for the carcinogenesis. The problem is we still understand very, very little about these processes and these alterations. One of the missions of PSOC and of Northwestern is to understand the processes better. And another cartoon that I borrowed from actually a textbook, it shows the structure of the genome of the chromatin. So, of course, you have DNA wrapped around histones, and then you have higher order chromatin structure, starting with a 30-nanometer chromatin fiber. Eventually, they all compress into the chromosome in the cell nucleus. So the problem is we know how chromosomes look like. We know the 10-nanometer chromatin fiber, how it looks like and what the structure is. We know a lot about histone interactions. Everything else which is shown in this and similar kind of cartoons is really our imagination, unfortunately. We know a high-order chromatin structure plays a critical role in gene regulation. It is important in carcinogenesis. The rest is really very poorly understood. So, and that's where Nanocymetry Core and the capabilities of it can potentially help. One of the instruments that is available is a multimodal nanocytometry instrument which combines techniques, label-based super-resolution microscopy such as STORM or structural illumination microscopy, so state-of-the-art label-based imaging, but it also combines label-free super-resolution microscopy, IsoStorm. I will quickly mention this later today. Live cell partially spectroscopic microscopy, and again conventional techniques such as confocal, total internal reflection, phase contrast, et cetera, et cetera. So really, the question is not even so much what kind of microscopy techniques are not co-registered by this instrument. The question is more like what kind of techniques are not co-registered by this instrument, because it pretty much covers every single technique, microscopy technique that would be of interest. And let me just talk about nanocytology then. As I mentioned, this technique can measure structure and dynamics of nanoscale at the nanoscale. It doesn't give you the resolution to resolve 20-nanometer objects. The resolution is 200 nanometers, but within each and every voxel of resolution, you can sense structure down to 20 nanometers and dynamics, again, at the nanoscale. You can work with live cells, no labels. Importantly, you can use very low light radiance comparable to the phase contrast. And this is important because most super-resolution microscopy techniques rely on very high irradiation intensities. There's always a question, are you inducing further damage or not? And with this technique, you can study live cells for days, basically, on end without damaging them. And it was co-registered with a number of other techniques. Let me give you a sense what this sensing actually is. This is one example. This is actually simulations. Imagine you have two organizations of chromatin. In this case, each of these balls is 10 nanometers. Basically, it's a nucleosome. And one of them has molecular correlation length of 20 nanometers. This one is 50. First of all, they look very, very similar. If you were to use even the best resolution possible, you would be hard-pressed to find the difference. But they are very different. There's 20 versus 50, very different. So it can very, very significantly affect transcription of the genes in these two configurations. Conventional microscopy can't really tell the difference. Everything looks very blurry and the same. At the same time, if you do pick up with US, if you do nanocytometry, from each and every you measure molecular correlation length from each and every resolution voxel, so you can really very easily tell these two configurations apart. So you don't image, but you actually sense. Well, in this case, actually imaging would not be even very helpful because it just these images look so similar. In the end of the day, you get images looking like this. Let's say this is a cell. It's one cell, helical cell in this case. There's a nucleus, there's a mitochondria here, and again, for each and every point, you measure the properties of the organization of the material, such as, let's say, the density or the molecular correlation distance, length within each and every voxel. You can image hundreds of cells at the same, or dozens of cells at the same time. This is a low magnification image. These red things are the cell nuclei. These are cells. The reason cell nuclei look different is not because they're stained with labels, but because the internal structure of the nuclei, of the chromatin, is so different from the structure of the cytoplasm. By looking at looking at the temporal autocorrelation, you can study dynamic processes, and you'll see an example of this a little later. One illustrative example, I guess. In this case, you started with the helical cells, labeled them with Hox dye, which is a very popularly used dye for live cell imaging, and then you activate the dye, and let's see what happens. So phase contrast says that nothing really happens to the cells, which is why this dye has been so frequently used for live cell imaging. At the same time, if you look at the cell nuclei here, before and after Hox activation, so this is actually without dye, this is dye and activation, you could see how dramatically nuclear structure actually changes. And this is really not surprising, actually, because if you were to look at H2AX acid, you could see accumulation of DNA double-strand breaks after the activation of the dye. So essentially, chop down DNA into chromatin into smaller pieces, which is what you see here as the loss of the overall structure and creation of smaller nanoscale strands of chromatin, basically, within this nuclei. In addition, there are dramatic changes in the dynamics. For example, mass flux almost stops. Almost any facilitative motion in the chromatin basically stops. Diffusion coefficient of chromatin changes dramatically. It actually goes up. You essentially stop the facilitative motion, and you have loss of Brownian motion of these strands of this DNA, which you chop basically. So it really illustrates two things. For one thing, yes, you can use PWS to detect, let's say, double-strand breaks, but it also illustrates how careful we should be when we use even what is widely believed to be live cell dyes, because many of them do cause damage, and we don't always know what damage has been caused. Like in this case, for example, if you were to look at the chromatin dynamics, you could see very dramatic differences. In the control cell, chromatin is highly dynamic structure. It moves all the time. At the same time, again, at nanoscale, at the same time, in a Hox stained cell, the chromatin dynamics essentially stops, and you have very different pattern. You can image live cells, as I said. Well, this was imaging of live cells. You can image cells doing something. For example, in this case, so this is going to be the cell, this is going to be the nucleus. We just add a little bit of glucose to the media, and you can see right about now glucose is going to come in. Okay, right, boom. You see this wave propagation. The structure of the cytoplasm changed, structure of the nucleus changed significantly. We don't really know what actually happens, because this process has never been observed before. But for the first time, using this technology, we can start asking questions of this kind. Another example of looking at now nanoscale dynamics, lamins have been implicated in control of nuclear shape and chromatin dynamics at the same time. And this is just an illustration how you can study these processes. These are wild-type cells, the cells expressing. So there's essentially underexpressing lamins. This is expressing lamins. And if you look at the mass flags, again, this is the cells. This is cell nuclei. These are cell nuclei right here. You can see that mass flags increases dramatically when lamins are are active. At the same time, diffusion coefficient stays pretty much the same. So lemmas don't tell you, don't control how fast chromatin, how fast dynamics happens in chromatin, but it essentially tells you how much. And mobile mass quite dramatically goes up as well. So more things in chromatin are moving. Again, for the first time, you can start asking this kind of questions and study this in live cells, again, for a prolonged period of time if you want. Another example is the work which is done by Tumor. One of the PSOC projects is focused on understanding how electrolytes or ions, ionic strength, or affect chromatin structure. And in this case, chromatin structure was perturbed by reducing intracellular potassium through using cardiac glucosides. And you look at the cell nucleus, you could see within 30 minutes, the chromatin structure goes from more a clump, which shows us a red color, to less clumped organization. So you can study this kind of processes. You can also look at how processes affect cell viability, or how the different processes change chromatin structure. For example, application of chemotherapeutic compounds. You can study which cells are going to survive, which cells are going to die, and look how chromatin facilitates these processes. You can look at individual cells and see how chromatin changes, as shown here. For example, cells with more heterogeneous, more compacted chromatin tend to survive chemotherapies versus the cells with a more homogeneously distributed chromatin. You can co-register PWS with techniques such as, something's not shown right here, but there was another image here. Trust me, as Donald Trump says. Trust me, there's another image here. It's a PWS co-registered with the STORM and user imagination. There's a beautiful image, which is not projecting here, the confocal microscopy. So if there are some need also to understand better chromatin structure in even more detail, even at smaller length scales. One of the questions, for example, is what is the structure of 50-nanometer chromatin fiber, or even does it even exist? It's been this is just an example of the kind of questions chromatin biologists frequently want to know. 50-nanometer chromatin fiber has been kind of a staple of textbooks, or even Wikipedia. If you go to Wikipedia, it's going to talk about 50-nanometer chromatin fiber as the key organization of the chromatin. The problem is, in the past several decades, there has been really lots of confusion. In some papers argue what the structure is. In some papers, some studies even say that the structure doesn't exist. It's an artifact of cell preparation, fixation for electron microscopy. So it's just an example of how little we know about the high-order chromatin structure. So that's where IsoStorm may become a very powerful technique to address this kind of questions. It has resolution down to a few nanometers, and it's a label-free technique. So potentially you can study processes without any perturbation to the cells. This is just one example. This is a DNA fiber right here. This is how conventional microscopy of this fiber looks like. You have to use labels to image it as well. So you have to stain it and then image it. That's how the image is going to look like. This is the image, the IsoStorm image. Okay, this is the IsoStorm image. You can see very, very fine details, which otherwise you would not be able to see using conventional fluorescent microscopy. That's the principles of super resolution. And in this case, you don't have to use any labels at all. This is a magnified view, and in this case, the resolution is about 6 nanometers. In order to validate this resolution, we couldn't really use conventional STORM because it couldn't even get to that kind of resolution. So we had to use AFM to confirm 6-nanometer resolution. Another example of IsoStorm, in this case, is the label-free imaging of mitotic chromosomes. You see that's how chromosomes would look like, and no labels, but that's how it would look like using without super resolution, and that's how they look like when you apply STORM. It's a magnified image of a chromosome. This is a further magnified image of the part of the chromosome. This is conventional fluorescence image. That's the power of super resolution. There is nothing that you can see here. Here you can see very complicated organization of the chromosome, and for example, this might be a fragile site. You can again start asking this kind of questions. And this technique again was developed as a collaboration between Hao Zhang's lab and in my lab, and now available for PSSC investigators. Last example of today is imaging of interface chromatin. That's how the image looks like, and now you can zoom in into smaller parts of the chromatin. You can study ask questions. For example, what are the sizes of the chromatin domains? As you can see here, the size of the domain as a function of DNA length is a power law, which is an interesting, essentially talking about, indicating fractal-like organization of the chromatin, which is a topic of significant interest after the discovery of the fractal DNA as a chromatin or DNA as a fractal globule, also by PSSC investigator Lina Mirny at MIT. This kind of studies can confirm and bring more light into the models that are developed by other PSSC investigators. Here you look at the real thing without any perturbations by the dyes or any other preparation. Other questions that can potentially be addressed is, for example, does the 13-nanometer chromatin fiber exist? Because for the first time, you can actually see the sizes of the chromatin fibers. You can see what's actually there, as opposed to function of fixation and preparation. Just to summarize, what this platform allows us to do is to go all the way from macroscopic tissues, 3 millimeters across, all the way down to nanometer scale and essentially individual molecules. So we talk about nanoscale sensitivity. There is no perturbation because you can work with live cells, tissues, and no labels. You can be sensitive to molecular, structural, and dynamical domain. The techniques are quantitative, and that's important for anybody who has done ultramicroscopy in the past. You can probably appreciate that it's really important to have quantitative information, which you compare one cell to another and you know what you're measuring. And it's applicable to complex systems such as cells or whole tissues. For example, how does the cell structure, chromatin structure, behave on a petri dish? It may be very, very different from how it behaves in a more complex structure such as QVAC or a extracellular matrix. Again, for the first time, we can start studying this kind of processes. Thank you. Questions? Okay. Well, any questions about Clinton's emails? No? Okay. No question. Okay. Thank you.
Thank you, Vadim. If anyone is interested in pursuing further collaboration or learning more about these techniques, you can go to the Physical Sciences Oncology website. It's just pssc.northwestern.edu, or reach out directly to Professor Backman. He's been very generous in collaborating with investigators across the university.
We'd like to collaborate, so everybody's welcome.
Yeah. All right, thank you. And next, I'd like to introduce Professor Andrew Mazar, who leads the Physical Sciences Oncology Center's Human PDX Tumor Core. Andrew has been engaged in some very groundbreaking work at Northwestern in establishing a PDX tumor repository. He has set up an amazing workflow with our oncology surgeons and working with the pathology core here on the Chicago campus. And he's been making tumor models available for collaboration to investigators, not only across the university, but across the country. So, Andrew, thank you for being here today. I know how busy you are. Yeah? All right. Are you going to need it? Okay, you've got the glove. All right.
Great. So, you guys hear me okay? Good. So, I'd like to talk to you a little bit about the patient-derived xenograph program that's ongoing at Northwestern, and also that is part of the PSOC grant that has been awarded to Northwestern. Thank Vadim for the first presentation, because between his presentation and mine, we kind of cover the whole spectrum of biology in my opinion in many ways. And so he starts sort of on the nanoscale, and the stuff I'll talk about will be more on the nanoscale, a bit closer to patients than it is to molecular, but we all kind of overlap at the region of cell and tissue, and so kind of where one starts to kind of blend into the other ones will we take over. And what I'm going to talk about today is the establishment of these patient-derived xenografts and some of their characteristics, and specifically from the perspective of how we use them for drug discovery and development, and also how they might be used clinically in patients, or at least the way they've been used to date. Maybe there's a lot of improvements to be made in that regard. Before I do that, I realized as I was walking in the door after I really loaded my presentation on here, I forgot to acknowledge people. So first of all, I want to acknowledge funding for this program, both from the Cancer Center as well as the PSOC grant, as well as the Lieber Award that came in from the Chicago Biomedical Consortium. And then really the work that I'll present here is very little bit is mine. It's actually my colleagues, and Glenn Kenny's here, Andre Volkov's another one of the people that's really done a lot of work here, as well as colleagues from University of Chicago, University of Illinois at Chicago. So this has really been a Chicago-based effort getting some of these models going, and some of the things that we've learned around the models. So with that in mind, as I said, just because of limited amount of time, I'll talk about what I know best, which is how we actually use these models to hopefully improve drug discovery, improvement, and development, I should say, of cancer drugs. And you've probably seen some variation of this schematic, and this is sort of the stepwise, if you will, iteration of how one develops or discovers and develops a drug. It's actually not true, because we don't do these things in sequential steps. They're very iterative and very cyclical. They tend to feed back on the previous steps as you work through this, but it does summarize a lot of the areas where we do touch on in terms of the techniques applied to actually taking a molecule or a target and credentialing that target in a particular cancer indication, moving it through that credentialing process, hopefully eventually getting a molecule that you can move toward patients and eventually getting into the clinic. And one of the limitations for successful oncology drug development has really been the utility of the models, and we've been sort of hamstrung for decades through the use of what we'll refer to as traditional cell line xenografts. So we'll show some comparisons and maybe hopefully some ideas of why the patient-derived xenografts might be an improvement over traditional cell line xenografts. But the bottom line is traditional cell line xenografts, as many people in this room probably realize, are not very predictive for even some hint of activity in the clinic. And so therefore using them to do discovery, credentialing is okay, but development is often not very useful, even though we still do it, people have done it historically, because you do know that if a drug doesn't work in a model, it probably will not work in man. But what you don't know is that if it does work in a model, it'll also work in man. So that there's a disconnect between those two predictors, if you will. A lot of the things that Vadim has mentioned earlier are things that could also be looked at with patient-derived xenografts. And so in addition to the tissues that come out of these models, it is possible to derive cell lines. And the cell lines, at least in the first few passages, do reproduce the clinical phenotype from which the patient-derived xenograft was established. I'll talk about that in a second. But for example, one of the things we're very interested in is developing drugs for resistant disease. And so we often get tumors from patients that have failed many different regimens of chemotherapy. And in fact, cell lines, at least in the early passages that are derived from patient-derived xenografts coming from those tumors, are often also resistant. Now, if you keep passaging them, like with many other cell lines, other than passage over the decades, they do eventually lose their resistance. And that's why the majority of cancer cell lines that you buy, for example, from ATCC, are pretty chemosensitive. I mean, they'll respond to pretty much anything, which is actually not true for the patient. But one of the beauties of the patient-derived xenograft program is, at least for a few passages in vitro and probably for more passages in vivo, we are able to recapitulate some of the resistant phenotype. And for a lab like ours, which is really interested in developing drugs for advanced cancer, which are typically patients that are refractory to many chemotherapies, if not all, or maybe even beyond chemotherapy, other types of therapies that are used in a particular type of cancer, we now have a new tool that can help. There was, obviously a question for several years as to how predictive these really are. And we'll talk about that in a minute, but what's been coming out of the literature over the last year is that, in fact, if you look at many models with many different drugs that have been tested, these models do actually have high predictive value for some activity in the clinic. That doesn't mean if a drug works in a PDX or patient-derived xenograft model, then it will eventually get approved. So I don't want to make that sort of leap of faith yet. But it does say that the majority of drugs that work with the patient xenograft models do show some activity in man. Really where we typically tend to use these models, in our lab at least, is sort of over here, lead optimization and development, but they can be used back here. We haven't really explored that a lot in our lab or within the setting of Northwestern, but these models are available for things like target discovery and target credentialing, which is labeled target validation here, as well as for asset development and screening. And again, these are available to all the Northwestern investigators and specifically to investigators within the PSOC. Okay, so very quickly, just an overview. I think there's really two salient features around these models. First of all, for patient-derived xenografts, these are models with only passage in mice. It's not to say we cannot make a concurrent cell line, which I've already alluded to, but for the patient-derived xenografts per se, we just take them from mouse to mouse. And so the way that these typically work, and I'll go through the workflow in a minute, but we typically will get a sample of a tumor coming right out of a patient. Sometimes this is a surgically resected tumor. Sometimes it's something different. For example, we can get effusions from patients with metastatic disease to the lung. We can get peritoneal dissemination, which usually comes as ascites in patients that have metastases or tumors that are disseminated within their peritoneal cavity. And for leukemias, we often get blood samples, so we get peripheral blasts and peripheral tumor cells that are in circulation. And any of these different patient samples can be used to establish a model in different ways. But once the model grows in the mouse, it is only passage from mouse to mouse and never sees plastic. And in doing so, at least within the experience that we've had internally, as well as the experience that's been published in the literature, is that at least histologically, and in terms of drug resistance profiles, the tumors do maintain the profile histologically. In other words, they resemble histologically the original tumor, and they also retain the drug resistance profile. Now, whether they are the same at the molecular level is an area of ongoing hot debate and hot discussion. We, at least myself personally, feel that sort of doing routine molecular characterization can be a little bit misleading, because if you do, for example, exome sequencing, and people have published on this, people can show that 98.8% of the sequence in a PDX, even after 20 or 25 passages, for example, in breast cancer models, is maintained as compared to the original patient tumor. But what does that really mean? Well, that means that 1.2 or 1% of that has changed. And say 1% is actually a lot of genes. And so are those important? Are they not important? We just don't know. And in fact, if you actually look at molecular profiling of many tumors are actually profiled with just 3 or 4 different genes being changed. And so consequently, we really don't know at the molecular level whether there's really integrity in going into multiple passages, but certainly at the histological level, and I would call it the phenotypic level, for example, drug resistance profile, we're able to maintain the original patient composition, original patient characterization. Sure.
Do you prefer to ask questions at the end?
Oh, you can ask any time.
So you take a sample to start PDX.
Yes. They're definitely heterogeneous. And in fact, one of the problems that we have in establishing that the patient-derived xenografts is we don't know exactly what we're getting from the patient. So in terms of the workflow, and I'll preempt myself a little bit, is that a tumor comes out of the patient and goes into the gross room, and the pathologist looks and he says it's okay, this is what we think it is. He gives us a piece of that, but what he gives us may not have been what he actually looked at. And in fact, we don't know that when we put it into the mouse. So we take and we formally fix a piece of everything that we get from pathology from down here, and the rest of it goes into mice. We cut it up into small fragments, put it into mice. But sometimes what happens is when you send that piece that we retained from what went into mice, and you do an FFPE, and you send it for analysis by a pathologist here at Pathology Core, they'll tell us, well, there's no tumor in that sample. Or your tumor, there's a few cells or whatever. And we see the same thing in the PDXs. In fact, we see examples where there's a lot of desmoplastic response, a lot of stroma, but there's not a ton of tumor. And this is indeed exactly what you see recapitulated in the patient-derived xenografts. And it's one of the problems when we actually set up therapeutic studies is because we're putting in different fragments into a bunch of different mice. Some of them grow really fast, some don't, some are in between, and it creates a lot of issues with control.
Tumors a lot of times they interact with normal tissue.
Right.
Edited by normal tissue. So when you take the sample from a patient, normal cells.
Right.
The moment you put it into the mouse, we're trying to, because this is the whole new realm of this is writing better than xenograph. But I just want to understand, you're now putting this mixture of tumor cells with normal cells in the mouse. You contain both of those type of cells.
Right. So the so-called the benign cells within the tumor are not really normal. They've been co-opted by the tumor, number one, but they're human origin when they come out of the patient. And they're probably human origin to some extent, at least in that first take. Okay. And this is not work out of our lab, but this is work out of other labs that's been published. But by the second or third passage, the human stromal cells and infiltrating cells, which are less so in our mice because they're NHG mice, but let's say the stromal cells, so endothelial cells and fibroblasts, are now mouse origin. However, those mouse origin cells are able to recapitulate many of the activities that the original human cells did. So this is the mouse stromal cells and the mouse endothelial cells seem to support tumor growth that can be activated in the same ways as we think the original human cells from the tumors were. And so, for example, the desmoplastic response, that is not a host response. It's really a response that's stimulated by the tumor on the host. And you can recapitulate that. We see this in pancreatic PDXs. We see this in our breast cancer PDXs. And I'll actually show you a picture of that. And, one thing I always stress, these are models. And if they weren't models, they'd be a human patient. And so there will be pitfalls to these models. But we're learning those as we go through this. But that being said, a lot of the original microenvironment components of these that are present in the original tumor are recapitulated in the mouse model, but they're mouse of origin rather than human origin. So at least that's what we know today. So, there's probably a paper in press right now that's going to contradict what I'm telling you, but won't be the first time. So everybody talked a little bit about the drift issue. And then I'll talk about toward the end of the presentation some of the data that we've tried to do focused on avatar studies, and some of you may have heard these, but this is actually using patient-derived xenografts either for doing what's called a co-clinical trial, where you're actually monitoring your patient-derived xenograft in parallel with the patient as they're being treated at a clinical trial, or actually trying to use the avatars for making predictions of clinical solutions for the patient. And we've done some of that work, and I can tell you a little bit about our experience with that, as well as how that might be improved going forward. So this is a slide that my colleague Vadim was kind enough to share with me. And this is really sort of talking just in schematic ways some of the different manipulations and different permutations that you might have with classical xenografts versus the mouse or patient-derived xenografts. And the only thing I want to spend this slide on is really to reinforce the idea that classical xenografts are really cell line cultures. So these are cells you can buy in a vial cells are quote-unquote homogeneous. We have found actually that even cells that are quote-unquote clonal cell lines that are around for ages, at least when you grow them in vitro, are not really clonal because you can actually get side populations of these things. And people have taken ATC cell lines and isolated side populations that they claim are stem cell-like, even though these are supposed to be clonal cells. So there's some active discussion going on around that. But the point being that classical xenografts are much more homogeneous for setting up models for monitoring tumor growth. In other words, easier to use is a better way to say homogeneous, I guess. Because you're taking cells and they're all consistent. You can put them into a nice suspension and you inoculate them into mice. As we've already alluded to, with patient-derived xenografts, we take fragments of tumors and put them into mice, and then you take that tumor out of a mouse, you grow it up to maybe a gram, and you cut it into 2 by 2 millimeter fragments, and then you reimplant them into next generation to expand, and you keep expanding until you actually get to the point where you have enough mice populated to be able to do a study, for example, to test a new drug. And there's a lot more heterogeneity in that. From a practical perspective, that creates the problem of actually trying to stage these animals, because we actually wait till our tumors grow till we see them, either subcutaneously or in the case where we do orthotopic models, we're able now to label these patient-derived xenografts with reporters, and we can follow them by imaging. But until we see an image, for example, in an orthotopic tumor, we won't start treating. The problem is they have 40 mice, and they keep coming up at different times. So that really creates a problem for the person that's doing the experiment, because they have to stagger when they start the treatment and when they end the treatments. These are things that we're able to overcome, but they're actually a lot more difficult to do with the patient-derived xenograft than they are with the traditional xenograft. This is something that came out from a review about 4 years ago, sort of showing on the spectrum of closest to human from farthest from human, how close the patient-derived xenografts are. Again, this data is a little bit dated, but if you can see that on the spectrum of things, the patient-derived xenografts are actually much closer to the patient with refractory cancer as opposed to the cell line xenograft. In fact, the majority of cell lines, as I've already mentioned, for example, are not drug resistant, even if you try to study drug resistance or they came from a drug resistant patient, because with multiple passages you lose that characteristic. The one thing that's very difficult to do so far with patient-derived xenografts is actually study them in immunocompetent mice or in models where you actually start studying agents that monitor immune surveillance, immune function, or drugs that modulate those activities. And that's really been a little bit of a challenge in the field. And of course, it's starting to come to the forefront because right now immuno-oncology is very, very hot. There's a lot of new drugs coming about in that area. And using patient-derived xenografts to try to test some of those drugs has been a real challenge. So again, a couple more slides because I've already talked about a little bit. This is using some of these models to develop or test in immunocompetent systems. And the approach that people have tried to take to make the PDX useful in immunocompetent models is to actually take a very highly immunocompromised mouse and reconstitute a human immune system. And this is problematic because when you do this, you actually have an immune system that's heavily biased towards lymphoid lineage, and very little is biased to the myeloid lineage. And so this is really not the same kind of immune system that you see in a human, and therefore anything that you test there that's an immuno-oncology type agent, so an immune modulator, will be suspect because it won't be able to modulate the immune system the way it would in an actual patient. And this is an ongoing area of study. People are now trying various ways of trying to bias the immune system back towards a more natural human system. But again, these are things that are very artificial and continue to be a challenge in the field. However, once we're able to set up a repository of patient-derived xenografts, and this is kind of where we're getting at with some of the tumor types that we have here at Northwestern. For example, we have quite a few examples of metastatic breast cancer xenografts, pancreatic xenografts, starting to build up a nice repository of bladder. Is you can actually do what are called xenotrials. And so this, in effect, is mimicking a clinical trial using patient-derived xenografts. So imagine if you had 30 patient-derived xenografts, each one comes from a different patient. And at the farthest extreme, if you wanted to, you could molecularly profile every single one of those and look for your target of interest if that's what you wanted to do. And you can actually randomize those animals. You can set up the models, randomize them, and then treat them just like you would a patient population to mimic your clinical trial. This is actually a very ethical and a much less expensive, believe it or not, way of actually testing a drug before it goes into humans. And this is work that actually came out of a review, which I think is probably the most seminal, not a of studies that came out of Novartis. I think there's about 73 authors on it. I think there's more authors than there is actual text in the paper. But basically what they did is they looked at 4,000 some different animals in these xenografts with 40 some different drugs. And in this particular set of studies, they predicted or were able to assess that the predictive value is greater than 80% on all these novel drugs and drug combinations. And so there's strong support from this paper and some papers that have come out since then that these models really are very useful in terms of predicting for clinical activity. If you think about it from the perspective of a pharmaceutical company, where your success rate at a preclinical stage is somewhere around 1 in 1,000, the ability to actually get 80% predictive values in an oncology drug, even if that's an overestimation, is still several orders of magnitude better than you can get right now. So this is really an advance in the field, if it actually turns out to be true across multiple drug combinations.
Build a repository of tumors to patient breast cancer.
Right.
How do you keep them? Do you freeze them?
Yes, we do. So I'll talk about that in a workflow, but the short answer is that in order for something to become a model, we'll call it a firm model. We've got engrafted models, we've got firm models. So it has to go through X number of passages and stay viable. And what we have to do is to be able to actually take the fragments, cryopreserve them, and then be able to revive them from cryopreservation. So you put them into fragments to cryopreserve the MSO, you bring them out, you take the fragment, put it back in the mouse, and it grows, you look at it histologically, it still looks the same. Only at that point is it a confirmed model.
So you need the passage to get to that stage, right? How concerned are you that you are already changing the original cancer that came from the patient, now you're going for several passages? Right? How different is that from the original one?
Yeah, so I mean, I'm sure we are. So histologically, they're not different. But, that's a pathologist assessment.
Right.
Molecularly, I am sure they are much different. In fact, on the two models where we have data, even in the first passage, they're radically different. But they look the same. And so what we think might be happening, but this is an NF2, so it's really a big thing, is that what we're selecting for is probably the most virulent subpopulation of cells within that called tumor. And so tumor, you said it's heterogeneous, and there's probably some small percentage of the cells that are some people call them cancer stem cells, some people call them tumor-initiating cells. Take your favorite phrase, but that's maybe what we're selecting for. And so in our very first GBM that we did, in our first glioblastoma, when we did the sequencing of the parent tumor, we had, predominant kinds of typical things you see in glioblastoma, like the bulk tumor, you would find the TCGA, but in the PDX, it's totally different. And in fact, what came up are things that were maybe at 1% penetration in the original patient tumor, which now became 20, 40% in the PDX tumor. But there were things like P10 mutations and things that you associate, ALDH type of overexpression, stuff like that you associate with the tumor-initiating cell phenotype. But I can't make too much out of it because it's just, NF2, but we've seen it in breast cancer, PDX, and we've seen it in the glial appendix. So I'm sure that we are changing it. But I think what we're doing is really selecting for the cells that, from a drug development perspective, we care about the most. Now, that may not be true for somebody that's trying to understand mechanism or other things, that those may not be the subpopulation of cells that they want from that tumor, but at least for our use in terms of predictive value for the clinic, I think the cells that grow out are the ones that we are most interested in.
Okay, so this notion of predictive value, you have this first tumor, you grow it up, and cut it up into chunks, as you said, 2 millimeter cube. Transplant it, and you have enough mice to do the experiment. In your experience, how much variability do you see with mice in the drug response? So you start with one tumor from which you made 20 different mice, all of them supposed to be identical, and then you do a drug response syndrome.
Yeah.
How much variability do you see?
So we have not to this point done that experiment in-house with our models because we have not had enough models to do that. Now there's two types of variability. There's one if you take the same PDX and put it into 40 mice, which is a little more homogeneous, as opposed to 40 different PDX from 40 different patients.
But that's a totally different question. Yes, we expect that software. You want to see the variability.
That's right.
But you want to be able to try
but that's true with tumor cell lines xenografts too. They sway, it depends, how who's measuring, what kind of moving calipers are in, that kind of stuff. But what we can tell you is that these models, when they read out something that is a real anti-tumor effect, and this to me is something that either shrinks the tumor or makes it go completely away in the model, it's really obvious. And you don't need a lot of mice to do it. You will see it with 3 mice or 4 mice. And so we've actually used that as a go-no-go barometer for us because we now say, unless it is just blasting the tumor away, we don't care about it. And I think this is, I don't know if this is the way pharma does it, but certainly as I think about my own projects, unless they have something that's that robust, I probably don't want to pursue it. It's not a completely satisfying answer, but okay. So I've already covered this a little bit. This is sort of the general workflow, and in fact, in the next 2 slides, I see more specific steps of how we actually go through this with both a solid tumor and a hematologic tumor. The one thing I want to point out is that we do get fresh tumors from surgery, but we often also get fluids. So for example, patients with metastatic disease have peritoneal dissemination, they have dissemination to their lung. Oftentimes when a patient has very resistant, highly metastatic disease to the lung, you get pleural effusion. So this is where the tumor actually breaks through the lung into the lining, which is the pleural lining, and starts to actually deposit fluid between the lung and your rib cage. Has to be drained out because it compromises pulmonary function. And that fluid from a patient with a pulmonary metastasis is full of tumor cells. And so what we typically will do is, as part of a procedure, it doesn't even require an RV, you just get the drainage, which can be as much as several liters of fluid, and you spin that down, and those are really highly metastatic, highly drug-resistant tumor cells. They grow like crazy in mice, and interestingly, histologically, even though those are metastatic cells, they look very much like if you go back to the patient's tumor 5 to 10 years ago from the original diagnosis for a breast cancer patient, it looks very similar to that. I'll show you one example of that. But it doesn't have to be surgical specimens. And in fact, one of the things that people have learned, and I think we're learning, is that the more aggressive the tumor is in a patient, the more aggressive it is in growing out in the patient drives xenograft. And in fact, this is usually a bad prognosis, although these are anecdotal observations. But if something grows in a patient for 3 weeks, it's usually a bad sign for the patient. If it grows in a mouse in 3 weeks, it's a bad sign for a patient. And we sort of confirmed this again anecdotally with our battery of pancreatic cancer drives xenografts, because the ones that grew most quickly, those patients did not survive very long, unfortunately. So again, I'm not going to go through every step. I show this more for illustration purposes, but this is a typical workflow for a solid tumor. In this case, I'm showing a glioblastoma. We typically will take the tumor that comes out of a patient, whether it's a little pellet of cells from a fluid, or whether it's surgically resected, as in this case. And oftentimes we'll put them in 2 places. We'll put them subcutaneously and either into the renal capsule, or subversa, or into the ovary, or interperitoneally. We do these interperitoneally, even if the tumors don't belong there normally, because it's a highly vascularized area, and the tumors will grow rapidly. So we want to kind of promote the ability of the tumors to grow as quickly as possible. And in fact, this is kind of a weird chimer, if you will, because we've got a glioblastoma that grew in the peritoneal cavity of this mouse. We had one of our neuro-oncologists look at that, and he kind of goes, well, that looks like a brain growing in the peritoneum of a mouse. And in fact, if you do the histology of this, it really does look like a true human glioblastoma, which I'll show you in a minute. And this just goes through the whole sequence of retransplantation and freezing. I don't want to talk about that either. The one other point I'll touch on is that with many of these solid tumors, we can actually mechanically disrupt them to make a tumor suspension. So the key here is not to use enzymes, but to mechanically disrupt it. If you make a suspension, that suspension will not stick to plastic. So it stays in suspension, and you can use lentiviral constructs that were developed here at Northwestern to essentially get 100% transfection with reporters. And then we can use these cells and put them back subcutaneous, solid tumor, even orthotopically, and follow them by imaging. So it's really a great tool to be able to do drug development stuff.
So you don't sort those cells?
You don't sort them. You just take the whole image, and there's a bunch of other junk in there too, just put it right back in. So other people will sort them, we have not. So.
And that's what you go in the serum. You have added serum to the cells.
I run it. We had serum. I don't remember. Yeah. so the second type of model that we do, we're doing a lot of leukemia models, and we can get both either biopsy tissue or basically other types of bone marrow samples, or we can get peripheral bloods, and we can get spin down cells. In this case, for leukemias, we'll inoculate intravenously, and these cells will actually home to the bone marrow, and they'll establish shop in the bone marrow. Again, you can label them, you can try to follow them, although we have not had a ton of successes for whatever reason, given the labeled cells are hard to see once we get into the bone. But these animals, they have a fairly rapid demise and progression and become moribund, and that's kind of when it's time to sacrifice. When you actually will euthanize these animals, you see a lot of the kinds of things you see in patients with advanced leukemia, which is splenomegaly, hepatomegaly, and in fact, the passage, you can isolate these cells both from the long bones, the femur sternum, as well as from the spleen. You kind of put them together, and then you re-inoculate IV, and they'll rehome to the bone marrow. So this is a way of passaging the AMLs. Now, this is really one where it's a question, is this really a patient-derived xenograft, or is this a cell line? Probably a cell line, because with sort of poor cytogenetic risk AML, you might have 8, 10 different clones, and you're probably selected for one or some small subset of clones that are probably most aggressive in this model. That being said, the model maintains the same drug resistance profile that the patient has. So, for example, this particular model, even though I don't label it, the patient fails cytarabine, and the model fails cytarabine. It just doesn't do well. So it's really a good place to do drug development studies. Okay, so some of the things that might be used to analyze pediatric characteristics. This is a whole host of things. We don't do everything here. Our approach has been basically to do some basic characterization, but then we leave the additional characterization to the investigator that might want to use it. We're happy to hand these out to people, but rather than do sort of a generic set of characterization that may not be useful to all people, we prefer to do sort of the basics, and then each person can analyze it in any way that fits their research program. And so in our shop, we will typically do some of the growth rates, some of the histological characterization, occasionally immunohistochemical characterization, where there might be some question regarding what the tumor is. One of the things that we've seen with some of these is because we grow these in highly immunodeficient mice, and these are samples coming out of humans, is we have overgrowth with EBV-driven lymphomas. And this is something that's been observed by others. We published a paper on it about a year ago, sort of a cautionary tale to the field about really making sure you characterize your tumors to know what they are before you start using them in studies. And remarkably, a lot of people don't do that. But we always do, and we've discovered that probably 30% of the tumors that we have that grow very quickly are actually lymphocytic tumors. And they're confirmed to be EBV-driven. This is something that you actually see in patients that are immunosuppressed or patients that are immunocompromised. So in humans, you tend to see these things grow out. And we recapitulate that when we grow these tumors in highly immunodeficient mice. If we grow the tumor, if we passage it into nudes, we no longer see the lymphocytic tumors. So that, again, is a way to either prevent it or to screen against it. And so it's something that we're cognizant of when we use these models. But by the time you get the models from us, we will usually have taken them through these quality control steps. We're also, whenever possible, doing the live to frozen and back to live model analysis, when we can revive the model from prior preserved specimens. But what we don't do routinely is mutational profiles or other types of molecular characterization or biomarker identification. Again, we feel that should be left up to the specific investigator to pursue if they so choose. So I'll spend about the next 10 slides or so just talking a little bit about the characteristics of a few of these models, as well as their use in some of our drug discovery projects. And so this is a pancreatic patient-derived xenograft. This is kind of what it looks like when it's grown subcutaneously. And we've got a number of these, and I just give up 2 examples histologically. A well-differentiated pancreatic patient-derived xenograft and moderately differentiated. Again, appearance-wise, these look nothing like a tumor cell line xenograft. These really look like true pancreatic adenocarcinomas. And this is the GBM that I showed you earlier. And this is just some different, let's describe this as a day in the life of this tumor. So basically, it was grown in cells and you put them into suspension and you put them intracranially and they grow in the brain. And so that's what this is. This is the intracranial lesion with the patient trying to xenograft from that very first intraperitoneal tumor that I showed you. And if you then take that and you image it by MR, you can see the tumor by MR growing in the brain. And if you look at it histologically by H&E, you see that the histology looks very much like what you see with many GBMs. It's a highly invasive histological profile. Looks nothing like the cell line derived gliomas. Cell line derived gliomas tend to grow as marbles. They're very circumscribed types of tumors, and you never see this sort of invasive phenotype. And in fact, in glioma patients, this is why patients recur and why they eventually die, because, the surgeon can remove, sort of the bulk tumor, but what they can't get are these little islands of invasive disease, and eventually those are the ones that grow back out. The other hallmark of this model is that when you put it intracranially or orthotopically into mice, is that these mice develop pretty severe cachexia, and that's actually an endpoint for our model. Even in the absence of being able to image, which we're actually able to do with these tumors, once the mice start to lose weight, they're really only have a week at the most that they survive. And so once they get to this point, you know that the tumor is basically overgrown the entire, or not the entire, but a large piece of the brain. And it's time to euthanize that mouse, whereas mice before tumors, continue to thrive. So this is an interesting endpoint for this particular model. Similarly, to distinguish between what we might see with the xenograft versus a patient-derived xenograft. So this is a traditional cell line xenograft. This is a cell line that's probably been passaged 5,000 times. It's been around in HPCC for, I guess, about close to 35 years. And this is actually a tumor line that was initially isolated from a metastatic pleural effusion from a patient at MD Anderson. A patient with triple-negative breast cancer that metastasized to their lungs. Whereas this is a ER/PR positive breast cancer that also metastasized to the lung, but this is one we created for a patient here at Northwestern. And again, if you look at the histology, what's typical with some of these rapidly growing cell line xenografts is they become highly necrotic. And what's not necrotic is just basically what I call a bag of tumor cells. I mean, this is all tumor cells here with this highly necrotic region. This highly necrotic region, by the way, does create problems when you actually assess these tumors in terms of drug response, because the necrosis actually is predominant. It's very difficult sometimes to see an anti-tumor effect when the tumors are this necrotic. As opposed to the patient-derived xenograft, you see a lot of desmoplastic response. You see a lot of what we think is probably fibrosis in here, and a lot of stroma interspersed among this area of tumor cells. So again, histologically way, way different. And histologically, the fact that this came from a pleural effusion, it really recapitulates what the original human tumor looked like. And this is, I think, the last example. This is just doing some immunohistochemistry around markers for colon cancer. And this is a colon cancer patient-derived PDX, where we've got the original patient tumor that expresses these two markers, CDX2 and CK20. And in the second passage, similarly, we're still maintaining the expression of those two markers. Again, not complete molecular characterization, but some of the main characteristics are being maintained in that tumor. So once we have these patient-derived xenografts set up, what we like to do with them is we like to set them up to be able to do drug development and drug discovery studies. And so I'll focus a little bit around the GBMs because we've got the most data here, but we've done this now with multiple models. In this case, as I mentioned, we're able to make the suspension and make the model or the tumor labeled with reporters. And initially, we grow these things subcutaneously. And the reason we run them subcutaneously is to develop enough tumor bulk to then be able to make enough suspension to populate a larger study. And so we'll grow something like 1 gram tumors, we'll dissociate them, and we'll start to inoculate them into the brains of mice to get these orthotopic intracranial glioblastomas. And these are the ones we actually do for our drug studies. And you can see this one lighting up by IBIS imaging. And this is, in fact, the kind of signal we like to see before we actually start treatment. So these are all animals with established disease before we start any sort of treatment. So again, trying to mimic the clinical situation. In this case, the clinical situation that we're trying to mimic is really in the recurrent setting. Most patients with glioblastoma get radiation, well, they get surgery if they're resection eligible, but then they'll get radiation treatment and chemotherapy, and then the tumors will grow back, and then you have to treat them probably with just chemotherapy alone. Patients usually don't get reirradiated, although that's starting to change. So this is really the setting where it's a recurrent tumor that's really pretty much resistant to everything at that point. So very quickly over the next 2 slides, we've done 2 studies. One in what is still a very sensitive model, at least with this drug. So this is a drug that's used for recurrent glioma, CCNU, which is also known as lomustine, and we've combined it with an inhibitor that we've developed in our lab called GSK3 inhibitor called 9-9-41. So this is a glycogen synthase kinase 3 inhibitor that we think is an enzyme that's involved in regulating chemoresistance. And if we inhibit it, we think we can actually sensitize tumors to chemotherapy. Now, in this particular model, the model is still somewhat sensitive to the chemotherapy, and you can tell that even in the monotherapy with just CCNU, there's still a fairly profound effect on survival. However, when you combine it with this GSK3 inhibitor, you get a much longer extension of survival. In fact, these animals would have kept going, and so we started sacrificing them to be able to harvest the brains, so we could slice through the brains and look for evidence of tumor, which, by the way, by the time you get out here, we can find no histologic evidence of tumor. And I'll show you that slide in a minute. But if you follow by IVUS imaging, you can actually see the progression of the tumor. And if you treat it, for example, with CCNU, it disappears, but then it kind of comes back. Now, we don't keep the treatment on board, just like you do in patients, forever. We treat for a certain number of weeks, and then we stop the treatment, and then we follow to see what happens after that. And in the combination group, the tumors disappear, and they really never come back way out here, despite the fact that you started treatment here. So this is pretty cool, but that's going to a resistant model. Yeah, so this is early data. We now repeated this quite a bit. But this is what I'm saying about if it works really well. So this is a study that only had an N of 5, and again, it's been reproduced now. But because the results are so profoundly different, the statistics are not even with N equals 5. And so in this particular case, this model is actually resistant to CCNU. So with CCNU alone, there's no effect. There's no effect with our drug, 9-9-41 alone. You combine it together, and again, these curves could go on basically forever, but we start sacrificing mice at each of these points again to harvest the brains to be able to look histologically for evidence of tumor. And again, you can see by the imaging in the CCNU alone group, there's really no effect on tumor growth. They just kind of keep going, and animals die pretty quickly. Whereas in the combination group, the tumors regress, and then they stay gone, at least by imaging. And again, it's a finite length of treatment. We stop and then we follow. And this is referring to the cachexia. So you'll see that in all the treated groups except the combination, you actually get pretty profound weight loss, which is correlated with the progression of the tumor by IVUS, whereas only in the combination group did the animals thrive. Now, if you take tumor sections from those brains, or sections of those brains, I should say, and do H&E staining, in the combination group, there's absolutely no evidence of tumor anywhere in that brain. And we've now gone by 5-micron sections to entire brains. So some of these things are 1,000 sections. I looked at every section and absolutely no evidence. And depending when you stop the model, sometimes you see these little, I call them vacuoles, but I'm not sure that's the right description for them. But you can see what the tumor used to be, where it's no longer there. But if you actually let the treatment go longer, these also disappear. So there's some promotion of regeneration of the neural tissue in the area where the tumor used to be in the combination treatment group. And just to show you that it's not specific to GBM, we've now done this in pancreatic, breast, mesothelioma, and AML. I'm sure for maybe one. And in all these models, we get combinations where we regress established tumors. In this case, I'm taking a patient trying to see about a pancreatic adenocarcinoma. And if you start the treatment when the tumors are around a couple hundred millimeters cubed, this is combined with cytidine, the tumors began to regress. We actually don't take these down to 0 because we want to harvest this to be able to do some histologic characterization of that tumor. And again, this has been reproducible with these combination treatments with this drug. And so I think I show you these slides basically just to reinforce the power, I think, of the PDX models in actually being able to develop new chemotherapy, new drug strategies for treating advanced disease. So finally, the last couple of slides, I'll just focus on what the clinical utility of these models might be. This is a collaboration that I'll focus on with Dr. White and his team at University of Chicago, as well as their GI oncologists that do pancreatic cancer treatments. And the so this is really looking at, I guess at this point it's been over 10 patients. I'm not sure exactly how many we have right now in the repository, where we actually obtained tumors from these patients and followed them during the time while they were being treated in the clinic. And there's a couple of things that come out of this that I think may help us think about what the next generation of this kind of approach might be. So you hear many people refer to these kinds of models as avatars because they tend to track the patient as that patient is going through treatment. And one of the ideas here is to not only be able to assess whether the available treatments might work for a particular patient, but actually being able to combine novel combinations. And the whole idea is that in parallel you would get molecular profiling on each of these patients, and you make predictions based on the molecular profiles as to new drugs and new drug combinations that might be active for that particular patient's tumor. So this is really a personalized medicine type of approach. And then be able to test them in the patient-derived xenograft before you actually put them into the patient. And the problem with that approach, at least in pancreatic cancer, is that there's no time because those patients are progressing so quickly that by the time the model grows and by the time you figure something out for them, they probably will have died 6 months before that. So we are trying to figure out ways to accelerate this process, and we're also trying to identify intermediate approaches, so in vitro approaches like organoids or microfluidics that can be validated using the patient-derived xenografts, but can actually eventually supplant the use of the patient-derived xenograft as an avatar. And so I will skip actually to the punchline in the interest of time. So the last couple of slides that I'll show. So this is one of these patient-derived xenografts. So many of the pancreatic adenocarcinomas that come out of patients, just like the GBMs, have primary resistance, which means that even up front you treat them and they don't respond to anything. And this is one of those patients. So gemcitabine used to be the first-line therapy in pancreatic cancer. It's probably not anymore, but at the time we started these studies, it was still considered first line. And that's what the docs would use. And you actually give this patient gemcitabine, and their PDX has absolutely no effect. But if you take that gemcitabine-resistant tumor and try some of the other things that people try second and third line in pancreatic cancer, they have various degrees of efficacy. And so FOLFIRINOX, FOLFOX, or FOLFIRI, slightly different amounts of activity, and gemcitabine and Abraxane, which was third line, is probably moving up, also probably has some reasonable activity in this particular patient. So this is more an assessment rather than a prediction. However, now looking at 4 different patients, so these are 4 separate PDXs, and instead of doing things sequentially like you do in a patient, we just decided let's take all the standards of care, throw them at the patient right up front and see what they do respond to and they don't respond to. And see if we can communicate that back to the clinician. And that's actually what you see here. You see that there's a high degree of diversity in each patient in terms of what their response is. For example, this patient did not respond to anything except for gemcitabine plus Abraxane. It's the only one that did really, really well. And to go back to your point, these are small n size. These are like 1 or 2 mice. But we'll do 2 tumors on each mouse. So you might see 4 tumors total. Other patients, this one pretty much nothing works. You maybe slow down progression a little bit, but not really. Maybe a little bit of something here, a little bit of something here. I think this just highlights, number 1, that these PDXs retain the difficulty of treatment that you see in the patient. And then number 2, trying to identify something that's active for that patient may actually help. Now, unfortunately for this patient, the clinician actually took this data and went back to the patient and said, this is what we saw, what do you want to do? And the patient said, give me this. So patient's choice, it wasn't the clinician's choice. So the clinician was not making a treatment decision based on these models. But unfortunately, this patient's disease was so aggressive that she passed before they could start anything. But again, these are just some lessons learned. In the interim, we hope to build on that as we take things forward. And I'll skip that slide. So with that, I'm going to stop and hopefully take some more questions if you guys have any. So, I mean, yes, it's going to core. So everything is in core. We have a repository. We have all the information we have so far.